And with that, I want to introduce both Ramesh Goel and Abeer Sohrab. They are going to be talking about benthic mats in western U.S. rivers, strain diversity, toxin risk, and risk alerts. So I'll
Thank
take it away.
you.
Thank you.
So my name is Ramesh Goel, professor, University of Utah, Salt Lake City. With me, my student Abeer, and then it's a whole group of people from SUNY, from Nevada, Reno, and from George Mason, collaborators contributing to this project. This project inspired by an incident in Giants National Park, Virgin River. Of course, the problem is prevalent around the globe. That's where our research started about these benthic pets. So this is an outline of overall presentation. We'll introduce benthic mats and the severity of the problem and some interesting results, mostly from genomic point of view we obtained so far, and then some nutrient acquisition pathways, why we know that nutrients are important in forming and supporting harmful algal blooms, but sometimes these blooms, they do occur in nutrient limited environment, and how do they do that, and how smart the cyanobacteria are. So these are, to start with, difference between planktonic and benthic. The planktonic cyanobacteria, they float, they are suspended in a water column, and these cyanobacteria with other phytoplankton, they grow with nutrients and other environmental factors. And these cyanobacteria, they tend to float. They go up and down in the water column, the planktonic ones, and because they have this buoyancy management system because of air vacuoles in their cell, so they can control the buoyancy and go up and down in the water column for nutrients. The benthic mats, instead, they form biofilm. Benthic means biofilm. They form biofilm on the surface, primarily on the stones or sand surface. In this context, when I say surface, we refer it as substrate. The surface which supports substrate, not in terms of organic carbon or food, but substrate in the form of implications are for the surfaces which support biofilm. And these, at the bottom, you see benthic mats. These are, okay. So see, forming thick mats, the bottom, and these planktonic suspended are formed in static waters, primarily in lakes, reservoirs, and these benthic mats are formed primarily in rivers. Of course, we do see benthic mats in lakes also, but generally benthic mats occur in flowing streams, lotic systems. Some pictures, these thick benthic mats right there, and then see this filamentous. They're so smart that they can extend outside. This is thick biofilm, but they can extend outside to acquire nutrient from the water column. So a bit about the research aspect when it comes to benthic versus planktonic. A lot has been done. When we talk about planktonic cyanobacteria, they're like, thousands of paper, manuscript reports have come out over last 3 decades. But as this picture shows, not much. Last 5 to 6 years, you start seeing publications related to benthic cyanobacteria. So there is not much information, and especially when it comes to the information related to this red fill ratio, carbon, nitrogen, phosphorus ratio, typical, which is a predictor and worldwide use for planktonic cyanobacteria, 106:6:1. So some pictures from Giants National Park right there. This formation of this toxic benthic mats is a worldwide problem. You see all over several places of the United States. Then you see, other parts of the world. Then in China, it's in Australia, and then in Europe also several incidences of these benthic mats. And this is from, not from Giant, but this rest of these pictures are from Giant National Park. This one you see is a water wall. It's a water wall. In Giants National Park, they have water diversion. They use to send some of the virgin river water to a nearby town, and that water is, of course, treated and then used as drinking water. So this is a water wall right on the walkway bridge, by the side of the walkway bridge, and you see this thick toxic cyanobacteria growing on this water wall. And you wouldn't imagine any nutrients there, and still they are growing with this in this clean water. So they're pretty smart, and then they can acquire nutrients, these nutrients, nitrogen and phosphorus, through different mechanisms. So different factors which contribute to much like planktonic cells, sunlight, nutrients. Bottom substrate means whether we're talking about sand versus gravels and stones, grazers. The most important question is the coexistence, the co-presence of other, not only other cyanobacteria, but other bacteria plankton, that means non-cyanobacteria. And what they do, much like in this room, we all are silent, but we have our own qualities, own attributes we carry. Likewise, the cyanobacteria, when they coexist in these mats, this thick biofilm, and these questions hold true for planktonic cyanobacteria, that how they synergize their effort with each other. Some cyanobacteria has some attributes, others do, and they complement each other. And this is something what we are going to highlight today for a specific genera, specific genus called Microcoleus, which occurs, I would say 8 out of 10 times you sample a benthic toxic mat, you will find this genus there present, whether it's New Zealand or Australia or Canada or Giants National Park or El River in California. This Microcoleus is present there, but it has special attributes how it survives. So these factors, so synergy between different cyanobacteria, how they synergize with other bacteria plankton in terms of nitrogen fixation, in terms of nutrient acquisition. So this is what happened in 2020. This Alaskan husky, I believe, went into Virgin River and swallowed some of these mats because they slough off, and within 20 minutes the dog died actually. And this was because of these thick mats were present in Virgin River, and that portion of Virgin River was stopped. So, and we know that we all go for narrows in Virgin River, we all enjoy. So how important and severe the problem could be. And that's where we immediately went and sampled Virgin River, and we found that this Microcoleus is the culprit, is present in Virgin River water and mats, and they produce this anatoxin and dehydrogen anatoxins. Some short of results from Giants National Park. You see right there, Cyanobium, Microcoleus. That's what these preliminary results. You see this is not rock, this is rock, but these all hanging thick biofilm, this is all Microcoleus. And in another picture, we saw that sometimes they appear deep brown, dense brown, sometimes they appear green, so they change color. And it's amazing to see that same genera, they could be brown, they could be green, they could be deep brown, and that is challenging. Just by seeing and by just looking at the morphology and color, you couldn't tell that it's Microcoleus or whether it's toxic. And other challenge is that toxic and non-toxic genera, Microcoleus, they coexist in same mat. So some produce toxins, others don't produce toxins, but they synergize their efforts in terms of nutrient acquisition and then synthesizing toxins. So, and that's what I was talking about, the collaborative effort of different cyanobacteria. So these are some of the questions we are answering. I summarize there that who those are, what are they doing, what are their synergies with respect to other cyanobacteria. And from a genomic point of view, sometimes just water sampling wouldn't tell you much, rather than, hey, my toxins concentrations are low, or I did not see dehydrogen, or why I'm seeing this dehydrogen. Because when I'm mentioning dehydrogen anatoxin, it's 3 times more as potent as anatoxin A, actually. So that's why it's very important to know whether a specific mat can generate these toxins or not. So with that, I would invite my student, Abeer, to walk us through some interesting results. Abeer.
Thank you, Dr. Goel, for the introduction. Now we will move towards more technical stuff, starting with the sampling area from where we collected benthic mats together with the team at Reno. We sampled 3 different river systems, Salmon River and Eel River from California and Virgin River from Utah. So we sampled from 2023 to 2025, but for this part of discussion, I will only focus on the samples that we have collected in 2023. So we sampled 3 different locations at Salmon River, covering the side channel, as well as the main channel. In Virgin River in 2023, there was a very high flow in the main channel, main river, and due to the extensive flushing, we couldn't see any benthic mats growing in the main channel. However, we did find well-established benthic mats that were growing in a side stream, which was flowing into the Virgin River. And we also sampled a pond, which was the starting point for that stream. So this location is at the starting point of the famous hike that is Narrows. So it becomes very important to study these sites as they are filled with people during the summer season. In Eel River, we sampled 4 different locations covering upstream, downstream, and also open and shaded regions. If we talk about the nutrient content, we see that the nitrogen and phosphorus both were low across all the 3 different river systems, and these rivers can be classified between oligotrophic to mesotrophic. So now we understand the nutrient context. Another big question is what these benthic mats are made up of. For that, we extracted DNA and did 16S sequencing. And so here we are looking at the microbial community at the genus level. And as you can see that all rivers, you see the most abundant band present, same band present everywhere, that is dark green. So it is related to cyanobacteria formedium. Now many species under the formedium genus have been reclassified as microcoelias. So we will stick to the new taxonomy, that is microcoelias. So you can see that all these rivers were highly abundant and the relative abundance was higher, and it was for formedium genus. And if we look into the Virgin River, we sampled different substrate, bottom substrate, rock and sand separately. So the mats from rock are denoted by RM here. The mats from sand are denoted by SM. We also sampled biofilms that were growing underneath these mats to know how these biofilms and mat are coexisting and helping each other. Basically, we see the taxonomic makeup is very similar in rock mats and sand mats. However, it differs a little bit in biofilms, as you can see here, the sand biofilms. In pond, we also see that other than the most abundant taxa that was microcoelias, we see another taxa here in olive green, which belongs to eukaryotic algae. And that makes sense because it is a pond and the water was stagnant. So a lot of eukaryotic algae was growing together, microcoelias. If we look into the Salmon River, we see the same pattern across different sites and across different time points. And also in Eel River, we see the similar observations can be made where the taxonomic makeup is mostly similar. However, the relative abundance vary from site to site and from point to point. One unique observation was that in Eel River, we found nitrogen fixing capable cyanobacteria, that is Anabina, growing here, which is represented by dark orange band. So it was found in some of the samples. So now we know that all these rivers have microcoelias, which is abundant in all these samples, but we don't know from which lineage does this microcoelias comes from. It is just one single species, or it is like multiple species that are growing together? And also we don't know how many strains or how many different microcoelias are growing together and producing toxins. So we dig a little deeper. And we studied the microcoelius amplicon sequence variants, where we find out that 47 unique ASVs of microcoelius was present in Eel River, 15 in Salmon River, and 27 in Virgin River. One interesting thing was that the common ASVs were only about 4, and those 4 ASVs were highly abundant across all these river samples, which are here highlighted in blue, red, and brown. Other ASVs, which are present in a lot of number, but they were relatively very low, less than 1%, and hence are not shown here. So through this, we know that microcoelius buildup or microcoelius community is exactly similar in Eel River and Salmon River and also in Virgin River. Now, another questions come up like, does this microcoelius community is like one single species, or do they have same toxic and non-toxic lineages? And what are their metabolic capacities are, and how do they function, how do they survive? And are these microcoelius community dependent on each other and counterpart for nutrient acquisition? So we did whole genome sequencing of pure isolated microcoelius strains, which we isolated from different regions and different creeks and rivers from Western US. So you see here, Eel River, we have 4 different strains, ER6, ER12, ER3, and R4. And from Klamath watershed, which is the same watershed as Salmon River, we have 2 different strains, RC16 and SR16 and SR17. And from Rock Creek, we have 1 strain, RC9. From Zions, we have 1 strain called ZRM 2.1. From Pine Creek near San Diego, we have SC15. And we also resequenced 1 toxic microcoelius, Microcoelius anatoxicus, which was classified in 2027 and characterized and named as Microcoelius anatoxicus from Russian River, just to use it as a reference genome. So if we look into the morphology of these strains, we see that the color of these strains vary from green to light brown to dark brown. So they look very different from each other. Even if you look at how these sheets or filamentous sheets are formed, some are thicker, some are dense, some are slimy, and some does not form sheets. Like for example, in RC9, it only have like a fluffy biomass, it does not form sheets. So these strains, if you just look morphologically, are very different from each other. Also, our LCMS results show that some of these strains were toxic, were producing toxins. For example, RC9, ER3, 4, SR16, 17, and R20, ZRM 2.1, all were toxic and others were non-toxic. So we want to understand now that just looking at them, we can't tell if these are different microcoelius or similar microcoelius and how do they relate to each other. So we did a genomic characterization where we built a phylogeny tree to understand how closely related these microcoelius strains are. And we used some reference genomes from 2017, non-toxic, and some toxic reference like Microcoelius anatoxicus PTRS3. And we see that our strain RC6 and ER6 and ER12 are very closely placed together with other non-toxic strains from the same region, that is California. And if we look into this cluster, the toxin cluster, we see that all of our genomes were closely placed together with Microcoelius anatoxicus. That means that it is the same species which is proliferating or which we isolated from different regions. Now, to confirm these results, we also looked into the ANI percent identity, basically average nucleotide identity, which tell us how similar these species are. If ANI between 2 genomes is greater than 95%, it means it comes from the same species. And here you can see a clearly distinct cluster between the non-toxic and the non-toxic. And we can say all the toxic strain belong to the same species, which are colored from light green to yellow, and the non-toxic toxic ER16 and ER12 were very similar. SC15, which was from Pine Valley Creek, is a new uncharacterized microcollier strains that we found near San Diego. So the basic takeaway is that all these strains from different river belong to same species, that is Microcollius anatoxicus. Now we will look just into curiosity, like benthic mats incidents have been happening in Europe, in Australia, and worldwide. So we wanted to know how are our strains or microcollias different from the global strain. So we put our strains into the global context. Again, we did a phylogeny analysis, and we took the other reference genomes, such as in orange, you can see the microcollier strains from Canada, and in light blue, you can see from New Zealand, and this one light green is from Switzerland. And two different clusters were formed, clade 1, which just belonged to non-toxic species, and clade 2, which belonged to the toxic species. And we can clearly see that our cluster of genomes from Western US was very closely placed together with the strains from Switzerland and New Zealand. And if we again look into the ANI identity, we see that the same cluster is formed between toxic and between non-toxic genomes, which suggests that globally, one single species, that is Microcollius anatoxicus, is responsible for benthic mass proliferations and also for toxins, which can lead to animal deaths, basically. So now the two question rises. These species belong to Microcollius anatoxicus, but how different they are in terms of producing toxins. Do they produce same kind of toxins? And does their metabolic capacity is just same, or they differ from region to region? To answer these two questions, and the biggest question, that do these species produce same kind of toxins, the answer is no. They produce different kind of anatoxins. So majorly they produce two kind of toxins, anatoxin A and dihydroanatoxin A. So this is the research done by our extended team, where we grow these 4 different strains, SR16, SR17, RC9, and PTRS1 over 40 days. And we see that in solid lines, it is anatoxin, and dashed line means dihydroanatoxin. So we see that 2 of the strains were producing a mixture of these anatoxins. However, RC9 and PTRS1 was just producing dihydroanatoxin. Now, why dihydroanatoxin and anatoxin are important? Like, what is the difference? And as Dr. Ramesh Goel said, that dihydroanatoxin and anatoxin does not differ too much if you look to the chemical structure, but they are 3 times more toxic than anatoxin A. So it becomes important to study like which species can produce which kinds of toxic, and which species is proliferating in our region. So we looked, so this is one gene, ANA-K, which is responsible for this conversion, or it is hypothesized to be responsible for this conversion. So we look into the anagene cassette cluster. So these anagenes consist of 10 genes from ANA-A to ANA-K. And we see that between all these genomes, the main code structures remains the same, which is from ANA-B to ANA-G. However, as we move, we can see a lot of accessory genes were added as we move from time to time and from distance to distance, talking about the geographical distance here. But more important is this ANA-K, which is highlighted in blue. The presence and absence of this can tell which toxic species will produce what kind of toxin. So we see that ANA-K was present in PTRS3, and also in RC9. However, it was missing from SR17 and SR16, which showed that ANA-K presence can lead to only dihydroanatoxin production. However, when NRK is missing, both type of anatoxin were getting produced, which also tells that NRK is not just one gene which is responsible for that conversion, but its presence can change the whole anatoxin into dihydroanatoxin. So now we understand that even though the species is Microcoelius anatoxicus, which is proliferating worldwide, it has the capacity to produce toxins, different toxins, and also at different time points. When grown in lab in same conditions, the toxin peak at different time points, as you can see in these graphs. So this is just lab conditions. In environment, these differences can be a lot large, at a large scale, because of various other environmental variables that are also included. So now we come to our second question, which is like one of the important metabolic pathway of producing vitamin B by vitamin B. So if you look into this flow diagram, we see, first of all, I will let you know what is vitamin B and why it is important. So vitamin B is a small element which is very important for the energy production in microcoelius. Also, it is responsible for running various metabolic core pathways in microcoelius, hence it is very important. But recent research say, and a lot of genes have, a set of genes is required to produce thymine diphosphate, which is an active form of thymine or vitamin B12 inside the cell. So previous research said that toxic strains does not have THY-D and are not capable of producing vitamin B. Therefore, they rely on other heterotrophs or on other cyanobacteria on their non-toxic counterparts. But interestingly, we found this gene present in our toxic genomes from Zymes, and also the strain we resequenced, because this time it was complete. So we also found THY-D there. Now to understand where this THY-D is coming from, is this getting horizontally transferred from other bacteria, or it is just contamination, or it was always there? We did a phylogenetic analysis of THY-D genes, and we see that THY-D from our samples is closely related to other toxic benthic mat, Oxalteria cyanobacterium, which tells us that THY-D was always present in the toxic lineage. However, some strains might have lost it in order to save energy to produce more toxins maybe. So they lost it over the time, and they might be dependent on other heterotrophs. But some strains, like from Zymes, are very much independent and can produce their own thymine. So now we come back to the same question, like, are these microbial community, which can help these mats, microcoelius mats, to proliferate, are same or not? So for that, we did a little bit of NDMS clustering, where we clustered the samples from our 3 rivers together and find that the taxonomic makeup is almost same in all the 3 rivers. Here is the graph from a recent research, which shows that the cluster differs when the most abundant species differs. So for Microcoelius anatoxicus, the cluster was always same, but when the dominant species was other than Microcoelius anatoxicus, the cluster varies. Also, we looked into the clustering from rock and sand samples, and we see here that rock mats and sand mats can be clustered together, but when we talk about biofilms, they form different clusters, meaning biofilms have different microbial composition, and they might be playing a role in the proliferation of these benthic mats. So overall, the takeaway from this slide is that Microcoelius anatoxicus form a tightly structured community, and this indicates that they have synergetic relationships with other heterotrophic bacteria. Now we will look into what relationships this might have, and this is our research which just got published in Science of Total Environment, which talk about how these mats acquire nitrogen. So we see that nitrogen was getting assimilated in Microcoelius mats through assimilation pathways. We also find some denitrifiers denitrifying the the system. However, that nitrogen was getting backed into the system through the expression of NIF, nitrogen-fixing genes. So we see a complete internal cycle, and this shows that internal mat cycling can be responsible for the proliferation of microcoelias in nitrogen-limiting conditions. Coming to another important nutrient, which is phosphorus. Now, we also looked into how the phosphorus is getting acquired into these mats. For this, we studied different kind of genes, basically focusing more on the organic phosphatase genes, because our conditions or our rivers were limiting in phosphor or inorganic phosphorus. So we look into phosphatase genes, phosphonate genes, which is another organic breakdown genes. Basically, we look into high affinity phosphate genes also, which are responsible for acquiring phosphorus in low inorganic phosphorus conditions. So here we compare two different groups, that is basically sand and rock. And this figure here shows that if the taxa is absent from that group, it is represented by white. If the gene is present, it is represented with black. If the gene is absent from that particular taxa, it is represented by gray. So we see that a lot of high affinity phosphatase genes like PST, ABC were found in sand samples, and a little bit we can see that the crowd was towards the phosphorus, alkaline phosphorus genes, which is 4A and 4D. And interestingly, one of the genes, which is GCD, it is a phosphorus solubilization gene, which solubilizes phosphorus from sediments or from the organic matter. It was more present in sand community rather than in rock community, as you can see here. So we'll look into now gene expression. These two groups have similar gene expression. Mostly they were dependent upon the alkaline phosphatase genes and high affinity phosphorus genes. But only the difference we see here is on phosphonates. Now phosphonates break the carbon and phosphorus bonds and acquire phosphorus through organic phosphorus, and this bond is very tight, so it's difficult to break, but this comes from the organic matter that might be getting decomposed on rocks. So now here we see that phosphorus is getting acquired through different metabolic pathways, and these microcoelous mats are capable to get phosphorus through different mechanisms. Now, at the end, I would like to conclude that we see that benthic microcoelous related dog deaths are rising, particularly across Northern America, which underscore a growing ecological and public health concern. The evidence from the Virgin, Eel, and Selwyn rivers suggests that there are similar widespread proliferations of microcoelous communities, and the only species which is responsible for this is Microcoelus anatoxicus, which is very adaptive, can produce a lot of different anatoxins, and also can depend on other heterotrophic bacteria for phosphorus and for nitrogen needs. So for the future, we're trying to build a model where we can predict these proliferations. Basically, we want to build a high frequency data set, more sampling, more data set, which can lead to a predictive model, including machine learning, so that we can produce advisories or mitigation advisory and how to locate these mats beforehand. So, but in case like a living room discussion could be, if you see this slimy stuff growing, where you go for swimming, just run. So at the end, I would like to acknowledge our National Park Service, people at National Park Service, at Science National, especially Robin, who helped us in sampling, and also NSF, which gave us the money to do this research, and also our partners at University of Nevada, especially Joanna and Dr. Rosalina from George Mason University. So that's all. Now we are open to questions.
You just talked about how these benthic mats are often growing in tandem with other organisms. Is there a trend that you can follow in those communities that you can see why the benthic mats are increasing in blooms as well as the algal blooms, or is it purely just benthic mats are now just being discovered to be more toxic and we're learning more about how they're doing that?
One of the reason is that they outcompete other cyanobacteria because of the adaptive strategies or how they acquire nitrogen and phosphorus. So if you walk into the stream, which is like a freshwater stream, nitrogen and phosphorus is very low. So you don't see much of the cyanobacteria, other cyanobacteria, mostly phytoplankton growing in there. So the indicator is basically carbon and phosphorus and nitrogen. So which could indicate that there might be benthic mats, but we can't see a specific trends. They can be present in some river system, but not present in another river system. And that's what we are trying to do. We are trying to collect extensive data sets so we can study how to predict these bloom and how to set up trends like, okay, this is going to happen this way. Yep. I hope I answered the question. Yeah.
Okay, if there's no other questions, let's give Ramesh and Abeer a good round of applause. We learned a lot.